The Shroud of Turin is this amazing relic. At bottom, it's a piece of cloth. It shows a very faint sort of image of a crucified man. And the crucified man calls us to remember Jesus' death and resurrection.
In 2015, a genetics lab at the University of Padua decoded the DNA trapped inside the Shroud of Turin and found genetic markers from China, India, Africa, the Middle East, and Europe, all encoded into a single piece of ancient linen. Not one of those regions, all of them, simultaneously. The scientists ran the data again, then again. The results didn't change. What they were staring at wasn't contamination, wasn't error, and wasn't anything the forgery theory could survive. It was a biological map of the ancient world, pressed into a cloth that millions believe wrapped the body of Jesus Christ after the crucifixion. The team stood in silence. Nobody spoke, because there was no clean explanation. Not for medieval Europe, not for the ancient Middle East, not for anywhere. The DNA pointed to a journey so vast and so precisely mapped that it broke every existing model of what this cloth was supposed to be. That finding has never been explained. And the deeper scientists have gone, since the harder it has become to explain anything else about it either, the face that wasn't supposed to exist. Before the DNA, there was a photograph, and the photograph already shouldn't have been possible. May 28, 1898, Turin, Italy. A lawyer and amateur photographer named Secondo Pia was granted rare permission by King Umberto I to photograph the shroud during a public exhibition. Pia hauled a camera nearly the size of a suitcase up onto scaffolding inside the cathedral, used magnesium flashes to light the dark interior.
He was wrapped in linen, and then his body was put in the tomb.
And exposed two large glass plates. Late that night, alone in his dark room, lit only by the dim glow of a red safety lamp, Pia lowered one plate into the developing chemicals. As the image materialized, his hands started shaking. He nearly dropped the glass. What was forming in front of him shouldn't have been there. On a photographic negative, light becomes dark and dark becomes light. The result is always distorted, inverted, unnatural to the human eye. That's how negatives work. That's how they've always worked. But what Pia saw on that glass plate wasn't a distortion. It was a clear, high-contrast face. Deep-set eyes, gently closed, a broken nose, a mustache and forked beard, bruising along the right cheek. For the first time in history, the figure hidden inside the linen wasn't a faint shadow. It was clearly a man, calm, dignified, yet bearing the marks of extreme suffering. The shroud's image on the cloth itself is already a negative. So when you photograph it, the negative of a negative becomes a positive.
They would want people to know, but I don't know why they wouldn't want people to know. Like, why am I convinced that they would hide that?
Well, if there is Antichrist on his way, and his goal, he already knows he lost. So his goal at this point is just to destroy as much as possible, get as many souls as possible.
A true photographic portrait, which means whoever created this image understood, centuries before photography existed, that the human eye cannot see the world in negative. They produced a perfect negative image with no way to view it, no way to test it, and no way to confirm it looked right. No artist in the Middle Ages could have done that. No scientist, no scholar alive at the time possessed the theoretical framework to even conceive of such a thing, let alone execute it flawlessly on a piece of linen. There was no camera, no darkroom, no process to verify the result. The human eye cannot work in negative, not then, not now, and yet the result is flawless. The shroud didn't behave like a painting. It behaved like a photographic plate, capturing a single frozen instant in time. That was the first crack in the wall of skepticism, and it raised a question that no one could answer. If a medieval forger made this, how did they produce a perfect negative image 800 years before anyone knew what a negative image was? They couldn't. That photograph was just the beginning, the DNA that shouldn't exist. In the 21st century, scientists stopped treating the shroud as a religious relic and started treating it like a crime scene. Genetics, spectroscopy, forensic molecular analysis, a biological data vault quietly preserving 2,000 years of history. In 2015, Professor Gianni Barcaccia's team at the University of Padua was granted unprecedented access. Using sterile micro-vacuum devices, they collected microscopic dust, pollen, and organic fragments from deep between the warp and weft threads. Space is sealed from the outside world for potentially thousands of years. They focused on mitochondrial DNA. It exists in hundreds of copies per cell, survives far longer than nuclear DNA in ancient material, and acts as a biological GPS for geographic origin and human migration. If anything had survived 2 millennia inside those fibers, this method would find it. Weeks of non-stop computer analysis. Millions of nucleotide sequences decoded and compared against global genomic databases representing populations from every region on Earth. Then the final maps appeared on screen, haplogroups, ancestral lineages, geographic origins plotted across continents. The lab went quiet. The findings, published in the journal Scientific Reports, demolished both theories simultaneously. If the shroud were a medieval French forgery, European DNA should have dominated overwhelmingly. If it were an authentic relic from Jerusalem, Middle Eastern DNA should have dominated. Neither happened. What appeared on screen was something neither camp had predicted and neither could immediately process. First hit, the Middle East. Haplogroups linked to the Druze, a historically isolated population in Israel, Jordan, Lebanon, and Syria, whose genetics have barely changed in millennia. A direct geographic fingerprint pointing back centuries. Second hit, Western Europe, haplogroups, U5b and H1 through H3, consistent with centuries of European handling. The Poor Clare nuns who repaired it, the House of Savoy who owned it, pilgrims across generations. That could be explained. Third hit, North and East Africa, haplogroup L3, pointing toward Egypt and Ethiopia. Contact with the earliest Christian communities on the African continent. Fourth hit, South Asia. Haplogroups M39, M56, and R8. Markers of the Indian subcontinent. Then the fifth hit, the one that changed everything, East Asia. Haplogroups D4 and G2A, associated with China, India, Africa, Europe, the Middle East, all encoded into a single ancient cloth. If this is pulling you in, hit subscribe, because this is the finding nobody has been able to walk back, and the deeper you go, the harder it gets to explain. A world written on cloth. No medieval craftsman in a French abbey in 1350 had access to genetic material from 5 continents. Globalization didn't exist, and no forger, however skilled, could have planted microscopic DNA traces from China and East Africa in a way that would fool genetic scientists 650 years in the future. That's not a theory. That's not even possible. The DNA on this cloth is a collective biological memory. When Barcacha mapped the data geographically, the distribution of genetic markers aligned with precision along a specific ancient route scholars had largely dismissed as legend, the Mendelian route. According to early Byzantine, Syrian, and Arabic sources, the shroud was folded so only the face was visible, a form called the tetradiplon, and kept inside a protective frame. This relic didn't stay in one place, and it certainly didn't begin in Europe. Its journey began in Jerusalem. From there it moved to Edessa in the 2nd century, where it remained for centuries at the crossroads of the ancient Silk Road. Caravans from China loaded with silk, merchants from India carrying spices, Persian diplomats, Arabian traders, pilgrims from every corner of the known world came specifically to venerate the image believed to protect the city. They stood close. They touched the frame. They pressed forward. And with every encounter, microscopic traces, skin cells, hair fragments, droplets of sweat settled into the fabric, layer by layer, century by century. The DNA of the entire ancient world accumulating in its fibers like invisible sediment. From Edessa, it moved to Constantinople in 944 AD, the greatest city of the ancient world, where every race and culture converged. After the Fourth Crusade looted the city in 1204, the shroud disappeared. It resurfaced in Athens around 1205, passed through French knights, and finally appeared in France around 1353 with the knight Geoffroy de Charny. The genetic distribution doesn't just match that route loosely, it matches it precisely, in the right proportions, from the right regions, in the right sequence. That is not a coincidence. That is a fingerprint written in human biology across 2,000 years. Pollen, blood, and a crown of thorns. The DNA was only half the biological evidence. The other half was written in plants. Professor Avinoam Danin of the Hebrew University of Jerusalem and Swiss forensic scientist Max Frey independently examined pollen trapped deep in the linen fibers. 58 plant species were identified, cataloged, and mapped. 17 were European, expected, given centuries of handling on the continent. The majority came from the Middle East, Turkey, and the Anatolian steppe, matching the Edessa to Constantinople route with striking precision. Then they found species that grow nowhere else on Earth, only in the narrow corridor between Jerusalem and Jericho. One species dominated above all others, Gundelia tournifortii, a thorny desert thistle. Its pollen made up nearly half of all samples and was concentrated specifically around the head and shoulders of the figure on the cloth. This plant, with its long, rigid needle-like spines, blooms near Jerusalem in early spring, precisely during Passover. It is exactly the species Roman soldiers could have twisted into a crown of thorns. A second species, Zygophyllum dumosum, grows only in the Judean desert and the Sinai Peninsula. Its pollen was also found in significant concentrations. A medieval forger working in France had no access to pollen from plants native. Exclusively to the region between Jerusalem and Jericho. Pollen cannot be painted on. It cannot be manufactured and applied microscopically across a piece of cloth. It can only be deposited through direct physical contact. It is a geographic seal, invisible, unfakeable, and perfectly preserved. The blood told its own brutal story. For decades, skeptics argued the reddish stains were ochre, cinnabar, or tempera. That argument collapsed in 2017 when Professor Giulio Fanti's team at the University of Padua examined the stains with transmission electron microscopy and Raman spectroscopy. At the nanoscale, the verdict was unambiguous: not pigment, blood, human blood. Type AB, one of the rarest blood groups on Earth, and the same type documented on the Sudarium of Oviedo. The cloth tradition holds covered Christ's face at the moment of burial, but the chemistry went further than blood typing. Within the samples, scientists detected nanoparticles of creatinine and ferritin bound directly to hemoglobin. Those specific concentrations occur under only one condition: catastrophic fatal trauma, prolonged torture combined with extreme dehydration and massive muscle destruction. When muscle tissue is repeatedly torn apart, a process called rhabdomyolysis begins. Creatinine floods the bloodstream in quantities the body normally never reaches. The blood on this cloth recorded that process at the molecular level, a biochemical record of agony that science now has a clinical name for. Over 100 wounds consistent with Roman scourging, leather whips embedded with lead weights and bone fragments, are visible across the body image. And there is one detail that has never stopped haunting researchers. The blood stayed red. 2,000 years later, it is still red. Old blood turns brown or black within weeks as hemoglobin degrades. This blood didn't. Analysis found extreme concentrations of bilirubin, a compound released by the liver during catastrophic physical stress, which chemically locks the red pigment in hemoglobin and prevents its breakdown for centuries. Not mysticism. Biochemistry. Precisely the biochemistry of a man tortured past the point of survival before the crucifixion even began. A painter can reproduce the appearance of a wound. No human hand can engineer the molecular signature of organ failure, rhabdomyolysis, and hypovolemic shock, least of all in the 14th century, when none of those processes had even been named. The test that got it wrong. In 1988, 3 of the world's most prestigious laboratories, Oxford, Zurich, and Arizona, carbon-14 dated a fabric sample from the Shroud to between 1260 and 1390 AD medieval, a forgery, case closed. The world accepted the verdict. Scientific journals cited it. Even the Catholic Church stepped back, referring to the Shroud as an icon rather than a confirmed relic. For most people, the debate was over. It wasn't. 3 decades later, researchers traced exactly where the mistake had been made. The problem wasn't the carbon-14 technology. It was the sample itself. The fabric came from the Shroud's most handled corner, a section that had been touched, folded, and repaired more than any other part of the cloth. Chemist Raymond Rogers of Los Alamos National Laboratory found something decisive. That corner had been expertly restored during the Middle Ages. The poor Clare nuns wove new cotton threads directly into the damaged edge, then dyed them with alizarin and sealed them with gum arabic to match the color and texture of the aged linen. To the naked eye, the repair was invisible. The main body of the shroud contains no cotton whatsoever. It is entirely linen. The 3 most respected laboratories in the world didn't date the shroud. They dated the patch. In 2022, physicist Liberato De Caro at the Institute of Crystallography in Bari tested the shroud using wide-angle X-ray scattering, a method that measures the degradation of linen, cellulose at the atomic level. Linen ages in a predictable, measurable way as its polymer chains break down under radiation, humidity, and temperature. The process functions as an internal clock built into the fabric itself. De Caro compared the shroud's molecular signature against a reference library of textiles ranging from Egyptian mummy wrappings dated to 3000 BC through documented medieval fabrics. The shroud's cellulose structure matched none of the medieval samples. It matched linen recovered from the fortress of Masada in Israel, a site dated between 55 and 74 AD 1st century, the time of Christ. In 1988, science declared it a forgery. In 2022, the atomic structure of the linen itself disagreed. The impossible image. One mystery remains that modern physics still cannot account for. And it may be the strangest of all. The image on the shroud was produced without brushstrokes, pigment, ink, or dye. Nothing was applied to the surface. The image exists only on the outermost 200 nanometers of the linen fibers, hundreds of times thinner than a single human hair. Under analysis, it is a chemical transformation, oxidation and dehydration of the cellulose surface, as if the fibers were exposed to an instantaneous, intense burst of energy from an unknown source. Scientists at ENEA, Italy's national energy agency, spent years attempting to replicate it. They tried acids, controlled heat, lasers, and gamma radiation. Nothing produced a comparable result. The only method that came close was a brief, precisely calibrated pulse of vacuum ultraviolet radiation. That technology does not exist in a form that could have been used in the ancient world, or in medieval France. It barely exists now in controlled laboratory settings. To produce the image across the full surface area of the cloth, a body would have had to release a burst of energy lasting less than a billionth of a second, powerful enough to scorch the outermost fibers, precise, enough not to penetrate or burn the cloth beneath. In 1976, NASA image processing analysis revealed something that stopped researchers cold. The shroud's image contains encoded 3-dimensional spatial data. The intensity of the image at any given point corresponds exactly, not approximately exactly, to the distance between the body and the cloth surface at that point. That is not a property of paintings. It is not a property of photographs. It is not a property of any known forgery technique. No artist, ancient or modern, has ever been able to reproduce it. The information encoded in that image requires spatial measurements that cannot be taken by a human eye and cannot be produced by any flat surface. Digital analysis identified shapes positioned over the eyes consistent with coins. Specifically, lepta minted under Pontius Pilate around 29 AD. These coins were in circulation for a narrow window of time in one specific region. A medieval forger could not have known this. Detail existed, let alone encoded it invisibly into a cloth image. The nails passed through the wrists, not the palms. Every medieval depiction of the crucifixion shows nails through the palms, because that is what medieval artists assumed. The shroud shows the anatomically correct position, which forensic pathologists only confirmed through modern research. Even the retraction of the thumbs is visible in the image, caused by damage to the median nerve when a nail is driven through the wrist. That neurological response was not described in medical literature until the modern era. No medieval artist knew it. No medieval forger could have painted it. Biology, chemistry, physics, botany, numismatics, forensic anatomy, every discipline arrives at the same coordinates. Jerusalem, between 30 and 33 AD. A medieval forger would have needed to produce a photographic negative 8 centuries before photography existed. Embed pollen from plants found only near Jerusalem manufacture blood, with trauma biomarkers that science couldn't identify for another 600 years. encode three-dimensional spatial data that no modern laboratory has successfully replicated, and deposit DNA from 5 continents onto a single cloth, from a French abbey with a candle and a paintbrush, with no way to see, test, or verify any of it. The Shroud of Turin is not a painting. It is a forensic record, a biological archive pressed into linen, a silent witness to a moment that altered the course of human history. The DNA mapped across its fibers traces a 2,000-year journey that no forger could have written, because no forger could have lived it. Here is the real question, and it forces a choice. The 1988 carbon dating said medieval forgery, dated to a repaired patch from a lab that didn't know the patch existed. The DNA, the pollen, the blood biochemistry, and the 2022 atomic dating all independently say 1st century Jerusalem. 2 verdicts. 1 cloth. Which evidence do you trust? And what does your answer say about what this is? Leave your position in the comments. And subscribe before the next video, because we found something in the burial record that makes everything on this cloth look like the easy part of the story.